We had discussed earlier the immune response against the tumor cells, then the evasion of immune response. Further, now we will talk about how to treat the tumor cells or tumors with the enhanced immune response when they are evading the immune response. We have seen the way the tumors evade the immune response. One, by antigenic modulation in the tumors, which confuses the immune system even if the antibody is produced. Therefore, the antibody fails to recognize the tumor because of the underexpression of antigens. Downregulation of class 1 MHC, with the result the tumor antigens are not presented to the immune cells, especially natural killer cells and CD8 T cells. Absence of co-stimulatory signals. Here, tumors secrete certain proteins which are inhibitory to co-stimulatory signals of the antigen presenting cells, especially the expression of B7.1 and B7.2 as co-stimulatory signals will fail to express, and without the co-stimulatory signals it is difficult for the antigen presenting cells to present the antigens to the immune cells. Then lack of high avidity tumor specific T cells. Now here the tumor specific T cells, even though when they initially show the immune response, they develop energy against the tumor because of certain protein factors which make these T cells inactive. Similarly, inefficient priming of tumor specific T cells. Here, in terms of presentation of the antigen is concerned, the antigen presentation becomes very weak as far as T cells are concerned. Therefore, T cells are not going to be activated. Hence, the cell-mediated immune response against the tumor fails. As well as physical and functional deletion of T cells may also take place because of certain tumor antigens or tumor proteins. With the result, the immune response totally fails against the tumor, hence the tumor succeeds in growing. Now, to overcome all these evasions, how to stimulate the immune response against the tumor and tackle it through immune mediated response. Let's see, there are about 5 different ways in which immune therapy against cancer can be worked out, and some of them are ancient and some are new. We will discuss each one of them because each one of them have played a major role in finding a treatment for cancer. These are the 5 different types of the immune therapies. One, non-specific stimulation of the immune system, active immunization against the tumors, that is by using tumor antigens, adoptive cellular therapy. Here, the tumor cells in fact are attacked by using the immune cells adopted from an individual. In the sense, it is a kind of ex vivo gene therapy. Then passive therapy with antitumor antibodies and cytokine therapy. Now let us see how each one of them work out against the tumors. So first, talking about non-specific stimulation of the immune system. It is a very ancient method and works very well as far as solid tumors are concerned. Here, the stimulation of cancer patients is done with an adjuvant, that is BCG vaccine. The BCG vaccine is a potent vaccine. It has got strong antigenic determinants and it induces strong inflammatory response at the site where it is injected. So the BCG is injected at the site of the solid tumor. Due to the presence of BCG in the tumor vicinity, it will induce the infiltration of the tumor site by a large number of phagocytes, especially the macrophages and the neutrophils. The macrophages being antigen presenting cells, since they invade the tumor area because the BCG is being injected there, they will start engulfing the tumor cells which are dead, as well as they will start processing the antigens of the tumor and start presenting them to the T lymphocytes, especially CD4 T cells as well as CD8 T cells, thereby enhancing the T cell response at the site of the tumor because of the presence of strong adjuvant, especially BCG. Similarly, adjuvant called as muramyl dipeptide, which is also derived from the mycobacterial cell wall, and this is also known to be a strong immunostimulant responsible for initiation of pro-inflammatory response at the tumor site. And this has also been used as an injectable material for the solid tumors to initiate the response of the CD4 and CD8 T cells. And it is found that because of this non-specific stimulation of the solid tumor site with the adjuvant, about 80% regression of the solid tumor can be brought about. And this can be used as an initial therapy before starting with the radiation therapy or chemotherapy of a person suffering from solid tumor, if the solid tumor can be reached at a site where it is growing, and the injection of the BCG can be done at that site, or muramyl peptide. Then coming to the vaccinations against the tumors, there are certain viruses that have been used which have been genetically modified, and their virulence has been completely deleted, but they become the carriers of important genes that can suppress the tumor growth. And by providing these virus with the important genes of the tumor to suppress the growth, these viruses can be used for infecting the solid tumors or tumor cells. It has been experimented with pox virus, especially by using pox virus as the carrier of the wild type P53 gene. P53 is a major and an important tumor suppressor gene. In majority of the tumors, the P53 gene is mutated, thereby preventing the tumor suppression. By introducing reintroduction of the wild type P53 gene, one can suppress the tumor growth because the P53 gene controls majority of other genes that are responsible for the immortality of the tumor. Therefore, the pox virus carrying the wild type P53 gene can be used for infecting the tumor cells and bring about suppression of the tumor growth. Similarly, a membrane-bound mucin, which is an important protein which can, when it is expressed, the tumor growth can be also suppressed. So P53 or mucin 1 gene can be used to be carried by the pox virus and infect the tumor cells and make them express the wild type P53 gene or mucin 1, thereby suppressing its growth. And this response is found to be transient response, does not last long, may give relief to the patient by suppressing the tumors for at least for about a year. Later, once again, the tumor may appear because these genes might have been modified or mutated after the introduction to the tumor. Similarly, same type of experiment was carried out with melanoma patients who were immunized with vaccinia virus encoding B7.1, B7.2 gene, or GP100 or A2 or melon A.A2 or tyrosinase, which are important antigens. B7.1 and B7.2 are supposed to be the co-stimulatory molecules of the antigen presenting cells. But if one makes these co-stimulatory signal molecules expressed by the tumor cell itself becomes the antigen presenting cell. And because it becomes an antigen presenting cell, because it already has its own antigens on the class 1 MHC, the CD8 T cells can easily recognize the antigen presented on class 1 MHC, and because of the expression of B7.1 on 7.2 by the tumor cell, the CD8 T cells can directly interact with the tumor cell, expressing that and bring about cell killing activity. So this approach has been found to be used for modifying the dendritic cells. So the dendritic cells which are harvested from the patient, in this diagram it is shown, the dendritic cells can be harvested from the patient suffering from the tumor, and then they can be cultured and grown in the tissue culture. At the same time, the tumor which is growing in the person also can be excised and it can be homogenized. The tumor which is homogenized will have all its proteins and antigens which can be added to the culture of the dendritic cells. By pulsing the dendritic cells like this with the tumor antigen, so to say, for example, the antigens such as MAGE, melon A, tyrosinase, or HER2 new, or mucin 1, one can trigger the dendritic cells to be activated, because all these are potent antigens, and these antigens, when they are processed by the dendritic cells get active, and when they become active, they can present these antigens to the CD4 T cells, and when they present the antigens to the CD4 T cells, CD4 T cells will become the responsible cells to enhance the immune response against the tumors. And this work has been carried out for patients with breast cancer, ovarian cancer, or melanoma. And using their dendritic cells, which are pulsed with these antigens, as shown in this diagram, the modified, or sorry, the stimulated dendritic cells can be given back to the patient suffering from the respective cancer, and suppression of the tumors can be brought about. And this is called as ex vivo approach. In this ex vivo approach, there is no genetic modification of the cell. The dendritic cells are not genetically modified, but they are grown outside and specifically stimulated with the antigens that are predominantly expressed by the tumor cells, as listed here with the blue font: MAGE, melon A tyrosinase, or HER2, or mucin 1. And these antigens are potent antigens. Somehow they are in fact hidden by the tumor cells and avoid the T cell recognition, but by making them to be presented to the T cells using a special approach like this ex vivo approach, by stimulating the dendritic cells with these antigens, one can in fact decrease the growth of the tumor or suppress the growth of the tumor, and it can suppress the growth for quite some time and give relief to the patient. So coming to other approach, the antigens that are very important for the tumor, which can be attacked by the immune cells, can be uh, made to be expressed, overexpressed, by using the DNA responsible for those tumor antigens, by injecting that particular DNA or the RNA that is responsible for transcribing these antigens or peptides into the dendritic cells. The same previous approach can be used. The, the dendritic cells can be harvested from the patient suffering from the cancer. They can be cultured in vitro, and when they are being cultured, at that time they are pulsed with the DNA or RNA, which is responsible for expression of those particular tumor antigens. And by making these dendritic cells process these DNA and RNA and express these antigens on their own, and then processed antigens are expressed by these dendritic cells either on class 1 MHC or class 2 MHC, will trigger the CD4 as well as CD8 T cell response against the tumor, which is in fact growing in the individual. So the approach which I had explained earlier, where the tumor cells generally do not express B7 molecule. B7 molecule is very specific as far as antigen presenting cells are concerned, and it is expressed by the activated antigen presenting cells. Now here a genetic modification is done, where the tumor cells are harvested from the person, and they are transfected with the gene of B7, that is co-stimulatory molecule. And by transfecting and expressing this gene using either a pox virus or vaccinia virus, they make this gene to be introduced into the tumor cells. The harvested tumor cells which are growing in the tissue culture now express the B7 molecule. They are inherently expressing class 1 MHC, and the class 1 MHC also is carrying the tumor antigens. And the tumor cells now behaving like antigen presenting cells, in the sense they will have the antigens that are specific to their own origin expressed by the class 1 MHC, and genetically engineered tumor cells expressing B7 molecule will attract the CD8 T cell response. The CD8 T cells immediately infiltrate the area where the tumors that are expressing B7 molecule as well as tumor antigens, and they interact with the tumor cells through their T cell receptor, and CD28 receptor interacts with the B7. That triggers CD8 T cells to undergo activation. The activated CD8 T cells will start secreting copious amount of IL-2 and proliferate in large numbers specific to the antigen that is presented on class 1 MHC. With the result, there will be an enhanced CD8 T cell response against the tumor which is expressing a particular type of antigen, as well as the tumors that are expressing now B7, and all the tumors that are expressing B7 molecule together with class 1 MHC are now attacked by the CD8 T cells, and they bring about instantaneous cytotoxic T cell response or cell killing, and the tumor cell expressing B7 will now be killed by the CD8 T cell response, which otherwise would have been avoided by the tumor because there was no expression of B7 by the tumor cells itself. So this is how one can bring about modified approach for attacking the tumors by triggering the expression of the antigens and making the immune cells pulsed with the antigens and activating them further in the laboratory condition, in the in vitro condition, and then giving back to the patient suffering from the tumor who has shown evasion of the immune response or immunosuppression against the tumor. Then another therapy that has been worked out, which has become very popular, is adoptive cellular therapy. Adoptive cellular therapy is nothing but harvesting of the immune cells from the tumor sites of the individual, and the tumor cells will have some immune cells which will be there in the tumor site. For example, natural killer cells which are there at the tumor site are called as LAC, LAK, in the sense they can be activated by the lymphokines. So therefore they are called as lymphokine activated killer cells. These are natural killer cells. Similarly, the CD8 T cells which are there at the tumor site are also called as tumor infiltrating lymphocytes. Probably these two cells are not showing an active and enhanced immune response in a person because of various tumor microenvironment factors inhibiting their action. The tumor microenvironment having different types of proteins secreted by the tumor may be inhibiting the active role of these natural killer cells and tumor infiltrating lymphocytes. Now, how to enhance these immune cells to attack the tumor of the person? It has been observed that the natural killer cells and tumor infiltrating lymphocytes, that is CD8 T cells, can be easily harvested from the tumor area of the person, and then by harvesting these to the LAC cells or natural killer cells, one can grow them in vitro under the influence of IL-2. Under the influence of IL-2, large number of natural killer cells proliferate, and these were already in fact at the tumor site. Say for example, they have been harvested from the melanoma patient from the melanoma area, and these natural killer cells which were harvested from the melanoma patient were already, in fact, recognizing the melanoma antigens. Now, by enhancing their growth by using the a lymphokine, especially IL-2, because natural killer cells respond to the IL-2 and they grow and proliferate. At the same time as they are proliferating, the homogenate of the melanoma tumor can be added to the culturing of the natural killer cells. The natural killer cells pulsed with the homogenate of the melanoma patient from whom they were harvested now have been now over triggered, and they are now active against the antigens of the melanoma patient. When they were inside the person, they were quiet, but now in vitro they have been activated. And such activated LAC cells, lymphokine activated natural killer cells, can be given back to the cancer patient through intravenous route, which will result in significant decrease in the melanoma of the person suffering. And it has been observed that there will be about 85 to 90% remission, and it is long lasting immune response, and the person can have relief for about more than 6 years. Similarly, the other cells, that is tumor infiltrating lymphocytes, it is observed that tumor infiltrating lymphocytes are nothing but they are CD8 T cells. These CD8 T cells can be grown in culture by harvesting from the person suffering from the tumor, and on triggering with IL-2 they can be multiplied in vitro, and when they are growing in vitro they can be in fact pulsed with tumor antigens, and tumor antigens, especially the class 1 MHC expression of the tumor antigens by the antigen presenting cells will make these CD8 T cells uh, to show enhanced immune response. It is observed that to the tumor infiltrating lymphocytes, when they are growing in the culture, if the macrophages carrying the tumor antigens are added with IL-2 stimulation, the tumor infiltrating lymphocytes get activated. It has also been observed that one can genetically modify tumor infiltrating lymphocytes by making them express certain specific cytokines. For example, one of the experiments that was carried out is to make the TILs express tumor necrosis factor, because tumor necrosis factor is generally suppressed in the tumors by the tumor genes, and because of the suppression of the tumor necrosis factor, tumors do not undergo necrosis. The tumor necrosis factor gene suppression is brought about by various other oncogenes. Now, to make the tumors regress, one can make tumor necrosis factor express in an area where the person is developing the tumor. So, tumor infiltrating lymphocytes, in fact, genetically modified with a gene for tumor necrosis factor, which is clubbed with a neo-R promoter. Neo-R promoter is obtained from the retrovirus. So neo-R is an antibiotic resistance gene. So neo-R will respond to neomycin by maintaining a small nanomolar level of neomycin dose in a person. One can make the expression of neomycin resistant gene expression promoter, and that promoter will make the tumor necrosis factor to be expressed, and because of the expression of tumor necrosis factor, the active tumors can be suppressed from growing. But it is observed that overexpression of tumor necrosis factor in large amount is also found to be toxic. Therefore, highly regulated at a nanomolar level expression of tumor necrosis factor is required to suppress the tumor growth, that is by genetically modifying tumor infiltrating lymphocytes. That is the second approach. The first approach was, I have already discussed, where tumor infiltrating lymphocytes can be pulsed in the tissue culture. They were harvested from the patient. As they were growing in the tissue culture, the macrophages of the patient obtained from the patient can be added to the tissue culture, and macrophages will present the tumor antigens on class 1 MHC, and under the influence of IL-2 you can trigger the growth of tumor infiltrating lymphocytes, and then those tumor infiltrating lymphocytes can be given back with the through the intravenous route to the patient, thereby decreasing the growth of the tumor. So then comes another approach where it is called as cytokine therapy, where you can modify the antigen presenting cells and make them respond to the expression of the antigens in an overwhelming way, or expression of the antigens in large numbers on class 1 MHC or class 2 MHC, so that CD4 or CD8 T cell response can be enhanced. Now here another approach will be to make the tumor cells as antigen presenting cells by making them express the cytokines that are responsible for enhancing the immune cell growth. One of the approach is by transfecting the tumor cell with GM-CSF. GM-CSF stands for granulocyte monocyte colony stimulating factor, which is an important cytokine responsible for triggering the growth of macrophages and other granulocytes like neutrophils, eosinophils, etc. So granulocyte monocyte colony stimulating factor gene can be injected into or transfected into tumor cells using a viral vector. And the tumor cells transfected with GM-CSF or genetically engineered tumor cells act as antigen presenting cells, and they in fact enhance the growth of antigen presenting cells of the immune response, especially because of the secretion of GM-CSF by the tumor cells. One can induce the large number of growth of dendritic cells as well as the macrophages. As shown in this diagram, the tumor cell which is transfected with the GM-CSF gene now expressing the GM-CSF in large amount, and that tumor cell which is genetically modified has been given back to the patient, and in the patient now the GM-CSF secreted will trigger the enhanced growth of dendritic cells as well as the macrophages. Now when these dendritic cells and macrophages get attracted to the tumor site because the cytokine is being secreted there by the genetically modified tumor cell, they will start now processing the antigens of the tumor origin, and they will present the antigens on class 1 MHC or class 2 MHC. And if they present, and as well as because since there is GM-CSF secretion actively going on by the tumor cell, those antigen presenting cells of the immune response, especially dendritic cells and macrophages, will also express B7 molecule. By expressing B7 molecule, which is the co-stimulatory molecule, their activity is enhanced. Now they are presenting the antigen on class 1 MHC as well as B7 molecule, which will interact with the CD28 of the cytotoxic T lymphocyte, and the class 1 MHC will present the antigen to the TCR of the CD8 T cells. At the same time, the B7 molecule also can interact with the CD28 of Th4, that is CD4 T cells or Th cells, and the class 2 MHC will present the antigen of tumor origin to the T cell receptor. Here you will see that enhanced activity and triggering of the immune response of both, that is Th cell response as CD8 T cell response, will be taking place because of the antigen presentation on both MHCs, that is class 1 as well as class 2 MHC. Such an enhanced response against the tumor will make the tumor be attacked by the cytotoxic T lymphocytes, and Th cells will enhance the response by secreting IL-2. So by enhancing the immune response using GM-CSF expression through the tumor cells, one can kill the tumor cells by this approach. This is also called as an ex-vivo gene therapy approach. Here, the tumor cells are harvested from the patient, genetically modified in vitro by using GM-CSF gene, and then given back to the patient. Therefore, it is called as ex-vivo approach, and that will be responsible for enhancing the immune response against the tumor. It is a kind of a suicidal response, in the sense the tumor cells themselves become responsible for triggering the immune response, and they are going to be killed. So the another approach will be the expanding the peripheral blood dendritic cells. The peripheral blood dendritic cells from the tumor patient, which can be harvested, and in the presence of GM-CSF, TNF-alpha and IL-4, they can be grown in the tissue culture, and highly proliferated dendritic cells which are proliferating in tissue culture can be pulsed with the tumor fragments. The tumor fragments are nothing but the minced pieces of tumor cells, and when they are pulsed with the minced pieces of tumor cells, the dendritic cells process these tumor cells and their antigens and be ready to present to the CD4 T cells. And when they present these antigens to CD4 T cells, the CD4 T cells are activated and they will start secreting the IL-2, which will be very, very important. The IL-2 plays a major role in activating CD8 T cells, and the CD8 T cells at the same time can recognize these antigens on class 1 MHC and then bring about cell killing activity of the tumor cells. So this is the other approach for bringing about suppression of the solid tumors using various different cytokines. The immunotherapy has also been carried out using the cytokines like interferon alpha, interferon beta and interferon gamma, which are used as immune enhancers and bring about suppression of the tumor, because these interferons are known to be facilitating the expression of class 1 and class 2 MHCs in the antigen presenting cells, which were underexpressed because of the tumor proteins in the tumor microenvironment. But by giving these genes as transfection to the tumor cells, or by using these genes to be carried by the tumor cells and express these cytokines, one can attack the tumor cells using the immune cells. Now coming to our most recent therapy, which is called as CAR T cell therapy. CAR refers to chimeric antigenic receptor of the T cell. Chimeric antigenic receptor in short form is called as CAR. It is nothing but a modified T cell receptor. Now how it is brought about? And this CAR T cell receptor therapy is used against B cell lymphoma only. It cannot be used against other tumors because it is specific as far as B cell lymphoma is concerned, and it has not worked out successfully against other tumors. So how it works against the B cell lymphoma, let us see. Let us see that the person suffering from B cell lymphoma whose blood is collected, as shown in the left hand side of the diagram, it is called as leukapheresis. After collecting the blood from the person, the leukocytes are isolated and the blood is given back. Isolation of the leukocytes and giving the blood back is called as leukapheresis, because here only leukocytes are isolated, but RBCs are not separated. RBCs are given back. So the person doesn't lose his plasma, the person doesn't lose his RBCs, but only few of his WBCs, some million cells of the WBCs he is going to be contributing, and that process is called as leukapheresis. Now after leukapheresis, the isolate the T cells from that leukocytes. T cells are isolated using marker identification technique using monoclonal antibodies. Now the T lymphocytes isolated from the person suffering from B cell lymphoma are cultured in vitro. When they are being cultured in vitro, now these T lymphocytes are going to be genetically modified using CAR gene. Now this CAR gene is nothing but it is a modified T cell receptor gene. Therefore, it is called as chimeric antigenic receptor. So, the T cell receptor, which is normally expressed by the T lymphocytes, is capable of recognizing the antigens presented by the class 1 MHC or class 2 MHC. In the case of Th cells, the T cell receptor recognizes the antigen presented by class 2 MHC. In the case of CD8 T cells, the T cell receptor recognizes the antigen presented by class 1 MHC. Now, without the presentation of antigen on the class 1 MHC, T cell receptor cannot directly interact with the antigen. It cannot directly interact. It needs to be presented antigen on class 1 or class 2 MHC. Now, CAR T cell receptor, that is chimeric antigenic receptor, is a modified T cell receptor which is capable of recognizing the antigen directly. That is the difference. Here, the genetically modified T lymphocytes, their normal T cell receptor gene is knocked off. It is shut off. Therefore, they cannot express normal T cell receptor, which is supposed to be recognizing the antigen on class 1 or class 2 MHC. It is knocked off. And the genetically modified T cell is made to express a modified T cell receptor, and that modified T cell receptor, that is chimeric antigenic receptor, is capable of recognizing an antigen called as CD19. The CD19 antigen is expressed by the B cell lymphoma in excess. B lymphoma cells express CD19 antigen in the form of a receptor on its surface on the membrane in excess. The CAR T receptor, that is chimeric antigenic receptor, which is capable of directly interacting with the CD19, is now made to be expressed by the genetically modified T lymphocytes. Such genetically modified T lymphocytes are called as CAR T cells. These are given back to the patients. The patient who was suffering from B cell lymphoma, from whom the T lymphocytes were isolated using leukapheresis. The T lymphocytes are genetically modified using CAR T cell gene, and their original normal T cell receptor gene is knocked off or shut off, and the genetically modified T cells are now expanded in culture and they are given back to the patient through intravenous route. On giving through the intravenous route, these CAR T cells now can directly interact with the B lymphoma cells, which are expressing an excess CD19 receptor. The CAR T receptor will interact with CD19 receptor, and especially these modified CAR T cells are nothing but CD8 T cells. And they bring about instantaneous killing of the B lymphoma cells. And this trade name of the CAR T cells is called as Tisa-Gen-Leclucel. Tisa-Gen-Leclucel as shown in the slide, and it is another trade name for this is Kymria. Kymria is the trademark name for the genetically modified CAR T cells. It has been approved by the FDA only for the treatment of B-cell lymphoma or acute lymphoblastic leukemia of the B cell, which expresses CD19 in excess, and it was approved in 2017 and has worked out successful in the patients in treating the B cell lymphoma. This is as far as some of the modified immunotherapeutic approach against the cancer is concerned. So we'll stop here and we will move on to the next immunotherapy in the next class.